So you might be thinking to yourself, I am here because I want to know more about the brain and how it works, and some of the neurochemical research that has been done and how it could apply to the classroom. So as a part of this presentation, I have three main ideas that I plan to encompass. The first revolves around exercise and how we can crank up our brains. The second involves whole brain learning and the nature of the learning cycle and elaborative encoding. And then the final idea that I wish to accomplish is the neurochemical influences that affect our emotions and our learning in the classroom. So one thing that is unique about our brains is that it actually makes up 2% of our body mass. However, the brain utilizes 20% of our body's resources, which makes learning sound like an intensive job for our bodies. Thus, one could imagine that our brain, just like any other organ, needs to be well fed and exercised in order to reach its optimal capacity, which leads us to our first idea of exercising our brains for learning. The brain is a very unique organ that performs a large array of functions and consumes an immense amount of resources. So how do we increase our body's ability to access those resources? Research by Medina suggests that you can crank up your brain by exercising 3 times a week for 30 minutes at a level 3 heart rate, which is comparable to walking up a hill. The exercise promotes oxidative stress by creating free radicals and nitric oxide in our bodies, which in turn influences angiogenesis. Angiogenesis involves the creation of new blood vessels in our brain, increasing its access to important resources like glycogen and oxygen. The mild oxidative stress causes stem cells to grow neurons and challenges your brain, which in turn gives your neurons practice at defending themselves from diseases like Alzheimer's and many others. Exercise also upregulates BDNF, also known as brain-derived neurotrophic factor. BDNF, in a sense, cleans out the nooks and crannies of your brain and allows for the synapses to form better connections. One thing worth mentioning is that the sedentary classroom environments that we typically use as teachers does not encourage the active benefits that come with exercising your brain. Ratey did a study that suggests that students demonstrate improvements in both reading and comprehension if they exercise, for example, run a mile a day before school, and they are in a better mood in comparison to students that take standard physical education courses. Reynolds uses an animal model and describes that studies using those models have suggested that when we exercise, our glycogen levels in our brains are for the most part depleted. However, after our brain has had a chance to rest and feed after the workout, our brain glycogen storage increases by 60% in the hippocampus, the prefrontal cortex, and slightly less in other portions of the brain, causing our brain to overcompensate for future work. However, if this workout regimen is continued, we find that the overcompensated glycogen quantities become the new normal for glycogen storage, which is thought to be involved with the development of a quicker and sharper brain. What this means is that if we want to encourage our students to be active, we need to design our classrooms in order to encourage that. There's so much that we don't know about these effects of exercise and what effects they have on the brain, except that it has significant health and learning benefits, and really no negative factors that we have found to this point, except that maybe your students may eventually outsmart you. The next area of research that I will discuss incorporates this idea of whole brain learning. How does it work? Students observe concrete experiences that creates sensory input, which comes into the portion of the brain generally categorized as the sensory cortex, towards the back of the brain. These observations are then filtered and sent to our temporal cortex, which is also referred to as the integrative cortex, and is involved with reflective observation. Our temporal cortex has a significant number of pathways going to our prefrontal cortex, which creates new abstract hypotheses, which is then actively tested by our motor cortex. After our motor cortex outputs information, then a new concrete experience can occur, creating new sensory input for the process to repeat. This complete brain experience allows for long-term potentiation to occur and help neurons to fire and wire together longer. As teachers, we can help students induce this whole brain learning and if done in conjunction with spaced repetition, which typically involves exposing students to small amounts of information 3 to 4 times in intervals no longer than 2 days apart, provides a support system that allows students to undergo elaborate encoding, especially if students are under a stimulating and an appropriate environment that's the right level of challenge for them. However, it is also important to consider that emotions play a significant role in students' learning. During the elaborate encoding process, it is important to consider doing business with the drug dealer in our students' brains in order to, as a teacher, create an optimal learning environment. The first thing to consider is our students' reticular activation system, or RAS, which seeks novelty and is easily bored. Our RAS desires a change of pace or something new after about 10 minutes, and if the RAS does not get what it wants, then it tends to shut off students' sensory inputs. Thus, as teachers, we should consider priming our students' RAS to promote student focus, which can be done by modulating your voice when presenting information, providing visually stimulating images, or maybe playing a song that relates to the lesson some way. Or another possibility is to ask students to make predictions to promote curiosity. The RAS is like a filter to our brain's drug dealer, who goes by the name of Amy G. Dalla, who wants novel information. However, if our Amy G. Dalla, or amygdala, is not interested, or potentially our student's RAS sends information from our sensory cortex that tells our drug dealer that they're being threatened, then the drug dealer will summon his goons and vasopressin will be released, which is a neurochemical that induces stress, anxiety, and fear, which is not useful if given in large quantities. However, if positive messages based on our concrete sensory experience are sent from our RAS to our drug dealer in our student's temporal cortex, then we are in a place to negotiate with the drug dealer, which is involved with our brain's emotional center and can initiate memory formation using pathways to our student's prefrontal cortex. At this stage, our students have two main pathways of what to do with information absorbed in the classroom. The less emotionally involved process is the pump and dump, where students retain information for a short period, usually a test, and then regurgitates it and then gets rid of it when it's no longer needed for test material. The second alternative involves the release of two important neurochemicals, oxytocin and dopamine, are handed out by the drug dealer, assuming he likes what he sees. Then dopamine is one of our brain's main feel-good chemicals that fuels our students' intrinsic motivation. Oxytocin, in simplest terms, is the opposite of vasopressin, and it's experienced by students as joy in their learning experience, and in turn it directly inhibits the fear and anxiety brought on by vasopressin. These positive neurochemicals play a significant role in helping students' neurons to fire and wire together for longer periods of time, which encourage students to take risks in their learning, and also assists in long-term potentiation, allowing students to elaborately encode information, and eventually these long-term memories are consolidated by our brain's hippocampus. As a quick recap, we have learned that if students exercise 3 times a week for 30 minutes at a level 3 heart rate, then the mild oxidative stress that our bodies undergo will increase blood flow to our students' brains, making it easier for them to gain access to glycogen and oxygen in their bodies, which is necessary for having a sharper and healthier brain. Then the idea of whole brain learning was explored, and how students take in concrete sensory experiences which processed by our RAS, then sent down to our amygdala and our temporal cortex, where memory formation can start and begin to release positive neurochemicals that allow incoming information to be processed by our students' prefrontal cortex, and finally tested in our motor cortex so new concrete experiences can be achieved. These new experiences are then reassessed through our sensory experiences. We also discussed the positive neurochemical effects that oxytocin and dopamine have in motivating our students, and how doing business with our students' drug dealer is an important part of being a good teacher. I appreciate your time in listening to this presentation, which has been brought to you by yours truly, Scott Clausen, and I hope you find this video useful in your future endeavors as a teacher.